hope to see you back next year. Thank you. And now biocomputer sciences. The methods of NLP are increasingly often used for tackling applied and fundamental problems in biology. Quite recently, you surely remember, an international group of scientists announced that they completely deciphered the human genome. They could get the genome sequence. So that is now available, but we can understand the meaning of only 2% of the entire area of the genome. So data scientists and bioengineers united to find the hidden meanings of the genome. Mikhail Burtsev will tell you more about it, candidate of physics and mathematical sciences, director of research at AIRI, the leader of DeepPavlov project, head of neural network lab at MIPT, and Olga Kardymon from AIRI.
Olga, hello. Great to see you again. Olga, just one question, to decipher the details, meaning the remaining 98% from 100, you will need your entire life for that? Well, I hope it will not happen like that, because we had such great progress in genetics and AI in the last decade, and I think it will help us to resolve this task as quickly as possible, much quicker than expected. Okay, we are looking forward to your report. Hi everyone. Our presentation is about GENA LM, the first DNA language model. But before we talk about our language model, let's talk about the construction of our genome machine. What is our genome consists of? What is DNA? What is a gene? And how the genes are working and regulated in our body. Then Mikhail is going to talk about our journey in terms of creation of GENA model and how we used it to resolve the real genomics tasks. And we are going to wrap up with the other applications of this DNA model. What are the real clinical tasks that we can resolve with it? And then we're going to touch upon the prospects of this technology. Now, many of you know that in 1990 there was a project that was launched. It was called the Genome of Humans. 10 years. Researchers used the then available technology to create the genetic code. In 2003, the project was closed, and it was basically thought to be completed. But now you need days to count the genetic code and sometimes hours to do that. Our colleagues before spent a decade on that. Now, what exists in our DNA? DNA is a sequence of nucleids. These are the letters, really. A, T, G, C. These are the letters. The RNA molecule is being read from that, and T is replaced with U in the same sequence. If we look at the genetic code, we will understand that by creating triplets of nucleative RNA forms amino acids, and amino acids is our protein. When it interacts with other proteins, with ligands and other molecules, it determines the most important things in our body. 3.3 billion base pairs, 3.3 billion letters. This is the weight, so to speak, of our genome code. And the Oxford researchers decided to print it in font size 4, 130 books. That's the library of our genetic codes with 78,000 pages. And that's a lot of information in our genome. But if you look at that in real applications in medical genetics, only 1.52% is used. This is around 3 million base pairs, 3 million letters that are being coded by the proteins in our body that make up genes. If you look at our library, it will be 1.5 thousand pages from 78,000 pages of our entire library. So what stands behind all this information, all this great array of information that is still in the unknown? This volume used to be called a rubbish DNA, and people thought that there's nothing there, but increasingly often we understand that there are some hidden elements in the DNA that regulate the work of our genes, and in this sequence, it is this sequence that tells us the mechanism of how the genome will work in one cells and will be switched off in other cells. So now let's think about the central theory of the molecular biology. The RNA are being read from the genes. In RNA, there are parts that code our gene, and there are some intermittent periods that are called introns. As part of splicing, these introns are being removed, and this matrix RNA remains, and this is used as for the further synthesis of the protein. It is interesting that when the genome was decoded, researchers thought that there will be around 100,000 genes in our RNA. Why? Well, because for quite a long time it was thought that we have at least 100,000 genes in our body, because we know at least 100,000 proteins in our body, but whatever we used to search through all that, we found only 20,000 genes. These 20,000 genes during the alternative splicing process can deliver 70,000 proteins, and when the protein has been synthesized, it can be modified additionally, and it can deliver another 250,000 proteins. So we have only 20,000 genes in our bodies. If you look at our genome machine in terms of how it works and how it's built, it's not a trivial process. First of all, for the RNA to be calculated together with the DNA, it should have a sequence. If you look at the surfaces, at the special distances, you can see the green zone, that is the gene. It starts with the part that is called promoter. This particular part is recognized as by the protein that becomes RNA, and the RNA synthesis ends at the end part. Around the gene, on the left and on the right, there can be enhancers. These are the parts of DNA that are linked with their own molecules, and together with the promoter, that they can strengthen the RNA. We also have silencers, and they work differently. If they receive the protein signal, they stop the gene, they disable it. Transposable elements are the parts of DNA that can jump from one part of the DNA to another part. If you look at that as as a text, it's like several lines that would jump from one page to another page, or to another part of the page. And sometimes the chains of the DNA are synthesized by RNA, but from this RNA the protein is never coding. This RNA has its own regulatory function, and this is all packed in a very highly structured and complex structured DNA molecule molecule. Obviously, this is a very high-level description. It does not give you all the peculiarities, but I just wanted you to understand how the machine works. It's very complex, and our genetic experts and biologists have a very hard time to decipher what happens in our bodies. The expression of the gene expression. How much do we receive of the end product from the gene? It can be protein, or it can be the RNA that does not code the protein. Here you can see the examples of the expression of the genes. The one is MIYH7 that is found in the muscular parts of the heart and the skeleton, but it is never used for the brain. The other gene, SLC1A3, is actively working in the cells of the brain and is very, very weak in the cells of other tissues and organs. And this example really shows us why, with the same sequence of DNA, depending on the different cells of our body, different tissues and organs are being formed. Why our tissues are so different? Because one genes are enabled, other genes are disabled. Now, how to define these functional elements that control the expression of the gene and the work of our gene? There are three approaches. The first is the experimental approach. It's the fixation of biochemical activity from a part of the specific part of the DNA. There are databases that researchers update by researching the parts that they research, and it is very but it's very expensive. It's very resource intense, and in the next decade we will not be able to make this database for all the cells, for all the cycles and states. The second approach is the evolutionary conservativeness evaluation, when the genome of a body is compared with the genome of other organisms. The parts that are similar are called conservative, and if these parts are evolutionary conservative, they are likely to have a very important functional role, and that means that you can make experiments with them further on and try to understand what role they will play. How do you check that? You can check that with the genetic approach. And for example, we look at a specific region, we implement some mutations with genome editing. It is done in the labs, and then we see how the functions of our bodies change, and by describing a mutation in one region in terms of the mutation, we'll be able to understand what was the role of this region. This is all very long, very expensive, very resource intense, and this is where our models come into play that were created with the deep learning. They can unite all these three approaches, and they can decrease the region being checked for our regions, and to understand their functional elements in these regions. In 2015, there was a disruptive research of DeepSea. It can assess one mutation in our genome, the change of one letter in a known coding element, and that can further lead to the removal of the transcription factors that regulate proteins, or can actually lead to its emergence. In 2019, the Illumina company offers a model that is also based on convolutional networks like the previous one. It can forecast the splicing. Splicing is the process when we have the matrix RNA, and RNA there are some coding and non-coding regions, and this process can be changed when we have mutations in these non-coding neurons. Now, before Splice AI, we had several tools that helped to forecast these negative consequences, but they were very low efficient. Splice AI really helps the informatics, the biologists, to assess the information and to make a conclusion conclusion whether they can change the function of the protein or not. In 2020, there is a DNA BERT that is based on transformer. That is a language model of DNA, and it is understandable that the transformer, compared with the convolutional networks, improve very much the forecast value of our experiments. 2021, DeepMind produces Informer model. It's a very complicated architecture with a transformer and convolutional networks, and it helps bioinformation experts to forecast the expression of the genes in our bodies. This has a very complex architecture. This is a very heavyweight model, and for the labs it's not really available. But at the same time, we see as an input of 100,000 pairs of 100,000 letters, which means that it improves the forecast. Now the trend has been set and understood. 2022, the biologists and IT experts and AI experts from AIRI publish GENA, a new model for DNA, and Mikhail Burtsev will talk in detail how the model was created and how it can be used for the resolution of the biology-related tasks. Thank you, Olga. It is my pleasure to welcome everyone at the conference. And I'd like to talk about the current neural network methods for the bioinformatics. We have heard about the successes that NNs were able to achieve in bioinformatics, but what will be next? What will be the area that will be the most promising in terms of ML for the analysis of DNA? And let's look at the problem of understanding of the attributes of the consequence of the sequences of the genome, and let's look at it as a problem of an analysis of a language. Recently, as you know, in terms of language processing, there was a great disruption thanks to transformer model, and based on these transformer models, a whole set of algorithms were built. The most universal model, most known one that is used for NLP is called BERT. Several years ago it was introduced, and it was the only one. Now we have more than 50,000 50,000 times it was cited. Now, why it is that efficient? It is it has transformer architecture. It contains of 12 layers with the on the input we have a text, and the model should look at the good representation, good understanding of this text. How can you achieve that? You can achieve that thanks to the use of the masked training. When we put the words on the input, we remove 10–15% of the words and ask the model to understand what will be these words. And this is how a model starts to understand better and better what words should be there on this places. And this training is called the training with the use of the masking model. The logical thing to be is to ask the following, that we were able to fill out the missed words. What would be the practical applications of that? You know, to understand the utterances and the texts written in natural language. The idea behind that is that today most successes were achieved thanks to the following. When we break down our models in two stages, first is the training done with the small samples of data. When we train this model to resolve universal tasks of restoring text, and then this pre-trained model can be used for the practical tasks. Now we can kind of transfer this knowledge of the pre-trained model to resolve specific tasks. For example, we add a small layer to our model and we train our model again to forecast the sentiment, the emotions behind the utterance, whether this utterance is positive or negative. The model tries to understand that. Also, on the input, we can have a question and the text with the answers, and the model should find the answer to our question in this text. This type of training helped these models to become the universal tool that can be adapted for many NLP tasks and for many tests. In many tests, NLP models are resolving the tasks better than humans even. Okay, then, so what are the implications for the bioinformatics? Well, it is directly related, actually. We, as people, we describe the world around us in a natural language. The evolution has described the living organisms with DNA sequences. That is, and this is how our phenotype is arranged. Why don't we use the same model that processes the sequence of the words in natural language to the sequence of the DNA that built up our genome? And this is how we can ensure the two-stage training. We can train the language model for the language of DNA, and then this good model of the sequences of the DNA can be used for resolution of some specific tasks in bioinformatics. And this approach was used 2 years ago when we took the same model, the BERT model, that is used for the natural language processing, and we fed the DNA language in it, and we trained the model to fill out the removed parts of the language. The name of the model was DNA BERT, and we found out that yes, this model can be used to resolve some specific tasks. In AIRI, we also think that this is one of the most promising areas in training larger and more advanced models for DNA. And we have started several months ago to develop our own model for the DNA sequences, and we called it GENA.
We have published the very first version of this language model, the version called GENA version 0. It is available in the Phase Transformers library. 610 downloads over the past month. So what is the difference between this one and the previous model, DNA BERT? With GENA, we use the tokenization that is commonly used with NLP in order to pre-process our input data, as opposed to the division that was used in DNA BERT. DNA BERT, we would take different input sequences varying in size, let's say with the length of 6, and then we coded these sequences with k-mers. We used a more optimal coding that yields a better result with NLP, the so-called BBE tokenization. This allowed us to increase the length of sequence that we can process with our model. DNA BERT could process 510 nucleotides, 512 rather. Our model could accept as an input up to 4,000 of those. We also augmented our training dataset thanks to adding data from the 1000 Genomes project. We also used the latest version of the reference genome, as opposed to DNA BERT. It once again gave us plus 10% in the number of data, the volume of data that we could use for our purposes, for pre-training our language model. As I said, training is always divided into two phases. Once we have pre-trained our model, we can apply it to different tasks, bioinformatic tasks. 4 tasks on the slide. In particular, the first one is predicting the sequence for promoter. We see that in this case, the model, the DeepPavlov model from the initial article is worse than our model, than GENA. The second one, DeepStar, is where you predict the enhancer activity in the genome of Drosophila. Here you can see that our models are once again different. With the GENA version, where we didn't have the additional data, is worse than the augmented model. The model with the data from the 1000 Genomes project is way better than the model that up to that point was the best for this type of tasks. We have also tested the pre-training, the additional training of our GENA model using the Splice AI dataset, a widely known dataset, which has been used for years now to develop, to fine-tune, to refine models. And in this case, even though we weren't able to beat the record, the best result, we came very close to it, and we spent much less effort than the community of experts that have been working on this task for many years now. Next task, the fourth one, DeepSea, predicting changes in the states of chromatins depending on alterations in single nucleotides. And here our model is comparable in terms of output, in terms of its results, with the existing model that has previously held the record. So we have additionally trained our model with a DNA sequence, but can we do something else? Can we do something even more advanced? Can we try to significantly exceed the current results in quality? We think we can. Why do we think so? Let's have a look at the models that are currently used for processing sequences. On the left-hand side of the slide, you see that the model guesses an input, a certain sequence, and tries to decode it thanks to allowing information to transport easily and to get accumulated. But the genome is much more extensive than that. It's much bigger than that. And very often between the sequence elements in the genome, the distance is way bigger than in the sequence that is used as input. With a transformer, we can put in tens of thousands elements at once. But with the genome, with the elements that are interdependent, such elements could be at a distance of hundreds of thousands between themselves. And unfortunately, that is a bit too much for the model. Another approach, recurrent architectures, is once again limited by the backpropagation, where you need to backpropagate several stages to process the whole sequence. Once again, the limitation of several dozens of thousands of elements at once. What we propose is this new algorithm developed at our institute called MIPT. With this algorithm, you can augment, you can add a special memory block to your recurrent architecture, and with this block you'll be able to memorize information that can be significant for other more remote parts of the sequence. The information in red, marked with arrows on the model, this information is necessary to predict an element in the future. This dotted line to the right, the dotted line in red. This means the red element is important for the element in the future. So, there is a question mark above this arrow. Even though the element does not fall into the observable field for our model, but thanks to additional training, that selects candidates for elements which may necessitate additional information, and thanks to continuous additional training and this additional block, memory block, we are able to process much longer sequences. We've already tested it with tasks such as copying longer sequences, classification of longer sequences. We are now applying it to NLP tasks, and we want to apply it to processing long sequences of DNA. We hope this will significantly improve the performance of our DNA language models. Also, we are planning to add to the training dataset not just versions of DNA sequences in humans, but also sequences which occur in other species, in other animal species, because we have a common genetic code, and there are a lot of concurring parts, concurring sequences, very similar. So we hope that by adding other species to the training dataset, our model will be better at handling tasks that are related to humans, but it will also be good at solving tasks for other species, bioinformatic tasks with other species. Of course, developing a model like that is not the final goal, is not the ultimate achievement. It is just a stepping stone for dealing with other tasks and problems in bioinformatics. We want to use other future versions of our language models for prediction of transcriptional factors, binding sites, for splice sites, and for many, many other tasks. Of course, we will not be able to cover the entirety of this ground, so anyone who's curious, who's willing to help, you are very much welcome to join in, to maybe share your ideas. How else we can augment our model and apply it to other bioinformatic tasks, and we hope that the model that we have now will also be helpful to you in different biomedical applications and in studying more deeply the way nature works. Thank you for your attention. At that, I give the floor back to Olga. Thank you, Mikhail. Fascinating. Thank you for describing the data science part of our model development. I would like to now maybe focus your attention on the practical applications of that model, the applications that are related to clinical studies, clinical purposes, anything related to the study of our genome, from sequencing the genome, from capturing the signal, where we need to identify whether this is an actual signal or this is an error on the dashboard. The convolutional networks can be very helpful. Secondly, the pathogen class. It is identified at the moment manually, but we still make good use of convolutional networks. We can also apply convolutional networks on the for identifying whether the mutation is a sign of a disease or not. We can also use GENA to figure out whether the metabolical path switches on, which disables the activity of a certain group of antibiotics. We can also predict the functional elements of our genome. We can predict the expression of genes and the elements that regulate it. So the range of purposes and applications can be very, very broad. Feel free to download GENA and apply it to your tasks. to your studies if you need any technical help with it. The Hugging Space and GitHub have all our contact details. At the Institute, we have researchers that work at the forefront of this scientific area, and we'll be happy to help you. We'll be happy to collaborate in additional training and augmenting our model for your narrowly specialized tasks. This is an area of our interest. We hope it is yours as well. And in conclusion of my presentation, I would like to talk about a thing that is not quite obvious. A lot of researchers that work on a model that can be applied in practice treat the model as sort of a black box. So there is this black box, we feed it some input data, and the output is predictions that you can work with. But what is inside the black box? Bioinformatics are trying to open the black box and study what's inside and study how the predictions are made. I am sure we can look forward to new breakthroughs in fundamental biology very, very soon, as well as in artificial intelligence. And it will be something even more significant than the breakthrough that we made with the peak prediction. Thank you. You mentioned the black box. I can imagine people tuning in just now, seeing you, the beautiful Olga, on the screen, talking about magic and a black box. Some may say that you are talking about things that are on the surface, but that's not the case at all. Also, I wanted to say that Mikhail came to science transitioned to science from studying it as a hobby, and you, Olga, you used to be a mountain climber. You conquered the Kilimanjaro peak. Fascinating.